So in this lesson, I wanted to kind of spend a little time talking about how cells can control the cell cycle we talked about in class. So remember, we talked about mitosis recently in class. Remember that the role of mitosis is for growth and repair. We haven't yet talked about asexually reproducing organisms, so don't worry about that right now, but do understand that it's about growth and repair. That after fertilization of, sperm and egg coming together, all the growth of an organism and all of the development is due to this process of mitosis. So it's a very important process. So there's tight control over cell division, and at times it may be accelerated or slowed or stopped altogether, and we talked in class a little bit about that. So because of this, the importance of mitosis and everything else, there are these checkpoints at various points in the cell cycle that halt the cycle if the DNA is damaged or if there are other problems. So I just wanted to go over this process called apoptosis, Apoptosis. That T there is silent. So apoptosis. If we have a healthy cell and there are some errors in DNA replication or some other errors, it's usually repaired. If it is not repaired and errors remain, there could be additional injury to the cell. If the cell is unable to repair the errors, the cell may undergo it's kind of a self-destruct process called apoptosis, and this is a normal control over the cell cycle. When a cell undergoes apoptosis from a normal cell, you start to see some condensation of the chromatin, the cell changes shape, the cell begins to fragment and eventually completely comes apart. That's apoptosis. And then a phagocyte, a white blood cell, will come along and gobble up the pieces and get rid of it. Now, the other thing I want you to know about are telomeres. Telomeres are at the end of every chromosome. This is junk DNA that actually protects the DNA from degradation, because with every replication of the DNA, some of the nucleotides are lost, and there's a change in the DNA as time goes by. So at the ends, there are these pieces of junk DNA that are the ones that are lost, and they don't matter. If the telomeres get too short, though, then that's actually a signal to stop cell reproduction because there's going to be damage to the DNA. But this is a normal consequence of aging. These telomeres shorten. So it looks something like this. Here's our chromosomes. These black pieces on the end are the telomeres. Over time, they shorten and shorten and shorten and shorten, and eventually they get to the point where cell division stops. So you see here, there's no more generations of cells being formed because the telomeres are too short. So that's one way that cell division is controlled. Now, there are some cells that are not limited by telomere length. These we call stem cells, and we'll talk more later in the year about stem cells, but one thing that stem cells have is an enzyme called Telomerase, which allows the cell to keep the telomeres long. So it keeps building more telomeres, and it's basically an immortal cell. So stem cells can continue to divide without degradation. They're immortal. Telomerase does some things. It inhibits apoptosis, which we just talked about. It regulates the cell cycle. It regulates gene expression, which we'll talk about later. And it can actually modulate how cells talk to one another. Now, remember we talked a minute ago about stem cells. Stem cells do a variety of things, including they reproduce themselves. They can become different things. They can fight off cell death or apoptosis. And they can reduce inflammation and swelling. So we'll talk more about stem cells later, though. Now, mutation. Mutation is an alteration of the DNA. If mutation affects the gene for a checkpoint of mitosis, this can alter the rate of cell reproduction. So we were talking about all these checkpoints. If there happens to be a mutation in a gene for a checkpoint, that's when you start to have a problem with cell reproduction. You've lost control. The cell has lost control, and reproduction sometimes proceeds without any break. So the cell doesn't rest, the cell doesn't enter interphase, it just keeps reproducing and reproducing and reproducing. Often, along with this, there's an alteration in cell communication. Sometimes the cells stop listening to one another. If you have a clump of these cells beginning to grow, this we call a neoplasm, and if they continue to grow and form a whole lump, that's what we call a tumor. So these are abnormal cells that have altered the way that they reproduce, and they're out of control. Now, this mutation that we talked about causes when the gene is changed to form what's called an oncogene. This is a cancer gene. It converts a normal cell into a tumor cell or a cancerous cell. Now, these genes that could potentially become oncogenes, the ones that have to do with cell regulation, regulation of the reproduction of cells, those potential oncogenes are called proto-oncogenes. And so we have isolated and understand certain genes that could become oncogenes. So here we have a cell with a proto-oncogene. Something comes in and causes mutation. It becomes an oncogene and leads to development of a cancerous cell. Now here are some examples of some proto-oncogenes just to give you an idea. One is called the EGF receptor. This is a growth factor in many cells. And so if there is a mutation causing an oncogene in the EGF receptor, that causes increased growth. There's also the BCR-A1 and 2 tumor suppressor genes. These are genes that normally keep tumors from growing. If there is a mutation in this proto-oncogene and it becomes an oncogene, these stop working so that they no longer suppress tumors. Another example is the HPV virus, human papillomavirus. It makes proteins that actually interfere with tumor suppressor genes, and so some people have found a connection between HPV and certain cancers. So here's an idea of kind of how this works. You have a mutation that inactivates a tumor suppressor gene, such as BCR-A1 or 2. The cells proliferate because there's nothing keeping them from doing that. There's problems with DNA repair genes. We have an oncogene now, and then mutation inactivates several more tumor suppressor genes until you end up with a tumor. Now, many of these neoplasms and tumors are benign. Benign means they grow slowly. The cells tend to stay together in a lump. But if these neoplasms are what we call malignant, they can be dangerous. And one example is skin cancer. Here is a benign neoplasm, and this is malignant melanoma. This is a malignant neoplasm. So let's talk about what causes them to be so dangerous. First of all, they grow and divide abnormally. They create dense layers of cells. They don't listen to each other. They don't care about any controls. They just keep growing. And typically they will show a very high amount of blood flow to the area because those cells need a lot of energy to keep growing. Secondly, they look weird. They're either missing chromosomes or have extra chromosomes. They have a disorganized cytoplasm. They look different. They look strange in their cytoplasm. And then third, the cells don't stick together. Okay? They can break away. They enter the bloodstream and float around. They migrate to other parts of the body. This process of leaving and migrating is called metastasis. So malignant neoplasms metastasize. So here we see some normal cells. You can see what they might look like under the microscope. They look normal. Have the nucleus, the nucleolus visible. Here's what a cancerous cell might look like. It looks very abnormal. The nucleus is extra large. It's got some strange clumps in the chromatin. And we can compare side by side normal cells. Cancerous cells have a large number of dividing cells. Normal cells do not. They have a large nucleus, small cytoplasmic volume, a variation in size and shape. Instead of all looking about the right thing, they're about the same. They have a loss of normal specialized cell features, so if a particular cell is supposed to look a certain way, they don't look that way anymore. They're disorganized in how they're arranged, and there's no boundary to the tumor. They just grow and spread. So what is cancer then? We say someone has cancer when the malignant cells have actually moved, they've metastasized, and they've invaded other body tissues and disrupted them. So their normal body tissues aren't functioning the way they're supposed to. Because the cellular changes, the DNA changes, and the telomere length and all of that accumulate over time, cancer rates tend to be higher as you age. That's just normal. You have a greater risk of cancer as you age. But you can delay or prevent cancer if you avoid substances that cause these mutations. These are called carcinogens, or sometimes they're called mutagens, these agents that can cause an increase of mutation. One example is cigarette smoke. Another is UV radiation, getting too much sunlight. Or there's certain chemicals in food. Asbestos is another example. So you can avoid cancer or prevent cancer by watching out for those things that cause the mutation in the first place. So probably more than you wanted to know about cell control over the cell cycle and cancer, but that's it for this one.